Okay, hello and welcome to this updated video in the Biotech Basics series. Today we're going to talk about gel electrophoresis, which is probably one of the most important biotech processes, absolute bread and butter stuff. Every molecular biologist knows how to do this, and you should hopefully get a chance to do this before you leave A-level standard. It is utterly common, very standard technique, but really important, and has given us such valuable insights into all sorts of areas of life sciences. Okay, so we're going to talk about gel electrophoresis, and I've given the idea there is high voltage chromatography. So if you remember what chromatography is, either having done it with photosynthetic pigments at A-level or way back at primary school where you were dropping water on ink and the ink was moving based on its density and its solubility, right? This is very much the same, but instead we're using electricity and a gel, not paper and moving water, okay? So you've probably seen pictures like this on all sorts of forensics programs. These are sort of barcodes here and scientists staring intently at them and going, look at these things. This tells me that the criminal was a lens grinder from Minsk. Go! Right? That's obviously not what happens, but you know, these pictures, these images have been taken up by forensics programs, CSI and all that kind of thing. And yes, they do show us incredible things, but in this lesson I'm going to teach you what these things actually show. So what is the genetic fingerprint? What is electrophoresis doing? How do we generate these pictures, and why should we care? Okay, so this is a little overview here. So at the top here, we've got a picture of a gel that has run. All those little bands there, they are fragments of DNA that have separated out from an initial sample. And down here, we can see, right, so down here, we've got the different tanks. Okay, for scale, this is quite a large one. And all these little dots here are all separate little experiments going on in this one tank. You can see this tank is full of liquid, it's full of an electrophoresis buffer, and it's got electrical wires, so it's got electrodes coming on either side, and the one nearest us here looks red, showing it's positive. So that's an electrophoresis tank. That's what this equipment looks like. So what is it? What is it for? Why do we care? Well, basically what you're doing is you're separating chunks of DNA based purely on their size. Okay, well, it's on their size, and it's used to generate these things are called DNA fingerprints and restriction maps. Okay, now you're thinking restriction, where we come across that, is that going to have something to do with restriction enzymes or restriction endonucleases? Yes, it is. If you spotted that, well done. Okay, now it relies on a really important bit, a really important aspect of DNA that will take you right back to the beginning of AS level, where we first introduced the nucleotide. Okay, now the nucleotide, as we know, a bit of revision, is made of a phosphate, a deoxyribose and nitrogenous base. Now this bit here, right, I will get this right eventually, this bit here, right, is the oxygen group, the double bonded oxygen, and that has a slight negative charge on it because of pairs of electrons just out here, okay? So a slight negative charge there. Now when you multiply that by huge numbers of nucleotides in one strand, overall DNA has got a negative charge, which means it will repel from other negative things and attract towards positive things. And this is where the electro part of our electrophoresis comes in. So to make these fragments of DNA move, we need an electrical charge, we need a potential difference to allow our DNA to move from the negative end towards the positive end, just like we have a bit of paper or whatever it is for chromatography. Okay, so first thing we do, right, but if we want to separate up DNA, is we need to actually cut it up. Okay, so we use restriction enzymes. Now remember what we said about restriction enzymes. They cut DNA at specific recognition sequences. Okay, so they look for the specific 3D shape of, in this case, is echo R1, GAA, TTCA, and if you go the other way, reading back, GAA, TTCA. So it's reading the same backwards as it is forwards. It's a palindromic region. So a palindrome means reads the same back and forwards, like the name Hannah, for example, right? So they look for these palindromic recognition sequences and they cut only where they find those sequences, okay? And they cut specifically there. Now DNA from different sources, from different organisms, different individuals, different animals, will have different sequences of bases, and it won't necessarily have the restriction sites in the same place. So if you cut up one sample of DNA with a particular restriction enzyme and a different sample of DNA with that same restriction enzyme, they won't necessarily cut up into the same chunks, okay? That's the sort of basis of how we're then going to separate out and get a different pattern for each individual. So let me show you what I mean by that with this fantastic use of PowerPoint animation. So we've got person 1's DNA here, person 2's DNA here. The black line is a strand of DNA. It's an analogous strand of DNA that we're looking at, okay? Now, person 1, because they are a different person to person 2, might have those recognition sites for this particular enzyme. Let's stick with EcoR1. Might have them there and there and there, okay? So you might find 3 recognition sites in that person's DNA. This person down here, person 2, you might actually find that they've got one restriction site just there, okay? And they've got another restriction site over there, okay? So this person, right, if I show you the whole strand here, only has 2 cut sites, okay? So first thing that's going to be different is if you take a strand of DNA, and I've got lots of props here, a strand of DNA that's a linear strand of DNA like this, and I cut it twice, I'm going to end up with 3 chunks, right? So if I cut that twice, I'm going to end up left with 3 chunks. Whereas if I cut it 3 times, I'm going to end up with 4 chunks. So person 1 would end up with 4 chunks of DNA, and person 2 would end up with 3 chunks of DNA. Instantly, I can see that they're different, okay? So this is what person 1 would end up with, okay? Those chunks there, okay? So this sort of medium-sized one here, a big one there, and then 2 little ones. And then person 2 would end up with this teeny tiny little fragment just here, right? Really tiny one, because that cut site was really close to the end. They'd then end up with this sort of quite big, sort of medium to big one, and that bigger one there, okay? So they've only got the 3 chunks, right? So instantly you can see the DNA is cut up differently because the restriction sites are in different places because their DNA is different, okay? Then once we've done that, so in our pot of sample, whatever it is from a suspect from a crime scene or from an ancient a sufferer of a disease or an ancient animal, we've dug out the permafrost, whatever it might be, we do that in a little pot. As you can see here, look, that's the sample. We've added the restriction enzymes there and we've given it plenty of time for the restriction enzymes to chop things up. And then we take the sample here and we place it in a well in the gel, okay, in a well in the gel. So that's one little sort of thing here, so one little sort of hole here. Now this stuff is what we call agarose gel. Not quite the same as agar jelly, but it feels quite similar in your hands if you've ever held agar jelly. So it's literally like a gel, a jelly. We boil it up, powder with water, we pour it into a mould, right, and it basically is a jelly. Now what that jelly does is it provides like a sort of an inhibitory environment. So basically, if you look at it under a microscope, all the sort of strands of gel are like a sort of really thick thicket in a woodland. I always think it was like a really thick hedge, okay? And the more agarose you add to your mix, so let's say you do 2, 3, 4 quite strong agarose percentage gel, those are going to be very, very close together. It's going to be very hard for the DNA to push its way through, even if you've got a positive and negative charge pushing the DNA away. It's going to be very hard. And obviously if you have quite a thin one, 0.5% agarose, then your DNA is going to move quite quickly. Now obviously it stands to reason that the smaller the chunk, the more easily it's going to be able to thread its way through, the more easily it's going to push its way through the jelly, right? So the smaller molecules, smaller chunks of DNA go further and the bigger ones stay back. So what you end up with is you end up with this lovely sort of laid out strands here. So you can see right down at the very end over there, we've got our lower molecular weight bits of DNA. And here we've got our higher molecular weight bits of DNA, okay? And they separate out. I always give the example in class. So this is, imagine taking a tennis ball, right, and a whole bunch of different species of dogs, from a Great Dane to a Jack Russell, and then lobbing it through a thicket of hedgerow, right? The first one to get there is going to be the Jack Russell because it can get through the hedge nice and easily. The Great Dane is going to struggle, okay? And that's very much like what we're talking about here. So we're using the gel to sort of inhibit the passage of the DNA, okay? Now to allow the electricity to pass through, you need to flood the sort of well here with a buffer, right? An electrophoresis buffer, often Tay buffer, right? Which basically is just water with a load of salts in. It helps conduct the electricity. Obviously, you put the DNA nearest the negative because, as we said, DNA is negative, so it's going to repel and it's going to try and push its way down here. Okay, I must remember where the limits of my box are so I don't lose my hand. And then after a certain amount of time, you stop and develop the gel. Okay, and look where the strands have got to. You don't want to leave them going forever, because then eventually what will happen is the little ones will drop off the other end and basically stick to your anode. So you've got to stop it at the right amount of time. Okay, so to give you an idea, look, that's where your negative electron goes into a well of buffer. You bung the sample in there, okay, and then you run your electric current, and the fragments will move through this layer of gel. Right, the smaller ones will go further, the bigger ones will go less far. And obviously it's a bit of a fiddle to get this, bit of a skill you've got to acquire to get the DNA loaded into the gels. You've got to learn how to use these fellas, right, these lovely pipettes. Okay, I don't know if you've ever used a dispensing pipette before, but basically, right, you have a special tip that goes on the end, like this. Now, it's always very important to keep the tip box closed as much as you can. So you have a special tip that goes onto the end. Now this one doesn't fit these tips, doesn't matter, but those are your disposable tips. You then would depress your plunger, suck up your sample and come over, and where the little arrow is here, you would lower this into the liquid, push your DNA sample in and then remove that. Okay, and these are obviously a molecular biologist's favourite tool. It's like a screwdriver to a molecular biologist, and the good ones have got this little button here, which allows you to, fire, just get rid of your tip without actually touching it, or indeed fire it at your mates in a university lab, right? We've all done it. So, but don't do it with anything dangerous in it, obviously. So, what happens then in the gel? So let's take a top-down view of the gel. So what happens then? So these are our fragments, as we've already talked about. So I've just basically taken those, copied those across from where we were talking about the digestion. So we've got our person 1, who cut up into that medium one, the big one, and the 2 sort of small ones. Then person 2 cut up into the teeny, teeny, tiny one there, and these 2 that are quite similar in mass. We'd then take that liquid and we would load that into, right, we'd then load that into the wells. Okay, so we're loading that into the wells there, right? So into the well here, right, and into the well there. We'd then apply the electric current, right? There's our strands, and they would separate out like so. So the red one might separate out like that. The 2 that were quite small, quite similar in size, would go quite far down the gel. The 2 bigger ones would stay a bit further back. And the green one would obviously go with the really little one, would go way over there, right? And then the bigger ones are sort of staying back here. Okay? So obviously not a perfect diagram. You can actually measure, right, when you do this with real DNA, you can actually measure the length of the strands from this information. I'll talk about how you do that in a little while. So if you run that together, look, there it is, and we stop turn our power off before our bit of DNA drops off the end. And that's a bit of a guessing game as to how long you need to get good enough separation without losing any strands off the end. Okay? Now, of course, you can't see the DNA in just with the naked eye, right? That would be lovely if you could, but DNA, you can't just see it. So once you've done this, you've got to actually develop the gel. So you take the jelly out of the tank, and then you've got to do something to it to make the gel, to make the DNA bands shine, or to make them sort of stand out. Now, the most common way you might do if you do this in a school laboratory is with a dye. Now, you probably wouldn't use ethidium bromide. That is a chemical that sticks very strongly to DNA, and as such, it's quite a potent carcinogen, right? Because obviously your cells will have DNA in them, and it will stick to the DNA, and obviously that damages the DNA. So it's quite a potent carcinogen. But something like Fast Blast, which is a blue dye, super safe, dilute it right down, dead cheap. Make a little trough, drop your jelly in there, and that will obviously soak in. The Fast Blast will stick to the DNA, and you'll be able to see that. Easy, cheap, doesn't give you the best resolution. If you've got 2 bands quite close together, you're not necessarily going to see them as separate bands. You can use radiation. If you use PCR to get your fragments before that you've done this, if you don't know what PCR is, go and watch the other video on PCR. You can actually use radioactive bases, and then they will glow, they'll develop a film, develop a photographic. So you then take your gel, put it on a photographic film, and the film will develop wherever the radiation was. You can also use fluorescence in the same way, but a really common way is to put it under UV light, because DNA reflects UV, whereas agarose doesn't so much. And that's what we're actually seeing down here, is basically shining UV light, which gives us some nice pictures. So I'll show you here a picture of a experiment that I did with some students previous year. Now they did get the order slightly wrong when they were loading their gels. So up here, look, these are the wells that they loaded the DNA into. And we've got our DNA, which has moved a certain amount. And we're basically looking for bits of DNA that match to show that they're from the same source. Okay. Now, as you can see here, row number 2, we've got this down here and this very light band down here. Now, it's typical for lighter bands to be lighter because they're less DNA. Pretty logical. And then over here, look, in this one, well number 1, 2, 3, 4, 5 cuts up the same. So well number 2 and well number 5 match. So they will have come from the same source. So if we're looking for, is this the murderer? Then we've got some DNA from a crime scene, we've got a DNA from a person, a suspect, and if they match, then it's the same DNA, okay? Now I told you could actually measure using this. These bits are a really common A-level question in the middle. This is called a DNA ladder. What we've done in here is we've put in fragments of a known size. So maybe this is 10 base pairs, 100 base pairs, 1,000 base pairs, 10,000 base pairs, 100,000 base pairs, right? Or whatever it might be. You could go up linearly, 100, 200, 300, 400, 500, or whatever is required for your experiment. And then using the DNA ladder reference, we can go, well, that one there is just a little bit lighter than this one here. So if this was 500, say, I could say that's about 490. Now you could graph the distance in millimeters that these went down your strand, down your gel, and make a calibration graph. Okay, and that's often an A-level question, so how can you do this? So you could do known points, so 500 went this many millimeters, 400 went this many millimeters, and draw a line, and then you could measure how many millimeters that went and use the calibration curve to get a pretty accurate estimate of just how big that strand of DNA is. But what we know from this, rough idea of how big these strands are, we also know that 2 and 5 were from the same source, okay? So that's basically how you'd interpret, but we'll do more on interpreting genetic diagrams or genetic fingerprint diagrams in the next lesson. So why are we doing this? What's the use? Well, individual identification, right? So identifying individuals, crime, relatedness. For example, if we're doing paternity testing, this is a really common way of doing paternity testing, right? Because you inherit your DNA from your mum and your dad, so therefore you also inherit your cut sites from your mum and your dad. So if your DNA cuts up like some combination of your mum and the guy you've been calling dad, then great, right? Then mum and dad. But if it cuts more up like a combination of mum and milkman, well, then there's questions to be asked, okay? And then you can see all that terrible daytime TV that uses this technique to work out paternity and relatedness, right? Usually it's used for paternity, can be used for maternity, although that's less often in dispute because she was usually there. So you can use it for diagnosis in terms of people with different diseases often have different cut sites. And one way that where genes are, how you locate genes that cause diseases, you take a load of people with a particular condition, a load of people without, do different restriction maps, and if there's a consistent difference between people with a condition and without a condition, well, it's likely that that fragment of DNA is actually to do with that disease, okay? Stands to reason. You then also have extraction for genetic engineering. So if you look back a few lessons, I told you we're going to skip over where we got this gene from, finding DNA fragments. We're going to skip over that for now and come back to it. Well, now it's now. So if, for example, you cut up your DNA and you found there was one fragment on your gel that you thought, that's really interesting, that must be the thing to do with this disease or this protein or whatever it is, you can physically cut that bit of gel out, dissolve away the gel, and just have that DNA fragment in a test tube. And then you can go into putting it into a plasmid, putting it into bacteria, running it through PCR, any of the other techniques that we've talked about, okay? So individual identification for crime is a good example. So if you find a DNA sample at a crime scene, then of course you might be able to use that to liken it to a suspect, but of course you need to have the suspect's DNA as well. And there's a lot of debate about whether there should be a national DNA database, but if you have ever been arrested for anything, probably your DNA has been kept on file by law enforcement, and there's a lot of debate about your right to be forgotten. How long does that stay? Does that stay forever? And also the idea that you can actually find out if a relative has committed a crime, because A lot of us are now voluntarily giving our genomes to Ancestry.com, 23andMe, all these other places that are sequencing our genomes. And then obviously if there's a criminal, right, and we find their DNA but we don't have a suspect, there's a question, can law enforcement use the big database that they've gathered to then go, well, I don't know exactly who they are, but they're related to this person, this person, this person. Ah, it must have been you, right? Are we happy with that? Don't know, right? So, for example, look here, I like to use this one. Maybe this here is a kid, right? This here is mum, and then we've got two potential dads, dad one and postman. And in this case, it clearly is dad one, because obviously you've got bands in common. Now, if you're given this, and again, more on this in the next lesson, as a question, what evidence is there? Make sure you talk about specific fragments. Make sure you say this fragment matches in the person and the suspect. Yeah, but as I say, more on that in a future lesson. So we'll end here with common exam tips and common misunderstandings about gel electrophoresis. Okay, the unit shouldn't take a genius to work this out, but KBP means kilo base pair. So one base pair is 1 A and T or C and G. Kilo is 1,000 of them, right? Just like 1,000 grams is a kilogram and 1,000 whales is a kilo whale, right? Kilo base pair. Just checking you're paying attention, right? Now, this is a difference in why I've got my string, and I've never actually found a better way to do this than with some pieces of string. Look carefully as to what the question is asking you. Is it asking you about linear DNA, or is it asking you about plasmid DNA? And I'll just do this briefly. If it's asking you about linear DNA, well, initially it's one fragment. Every cut adds a fragment. So basically, if you've cut it once, I've got two fragments. So if I see two fragments on a gel, it's been cut once. If I see three fragments on a gel, it's been cut twice, right? So fragments equals cuts plus one, right? So that's been cut twice, it's 3 fragments. If it's cut 3 times, it'll be 4 fragments. If you, on the other hand, have circular DNA, like a plasmid, like this, if I cut my plasmid once, well, it's still 1 fragment, right? So I cut it once and it's still 1. If I cut it twice, then it's 2, okay? So read the question and be sure what type of DNA you've been given. Is it linear DNA, like human DNA, or is it a plasmid, right? Because that's important. Now, please bear in mind there are thousands and thousands and thousands of copies of each piece of DNA. So if I get my fragments here back of my bit of DNA that I just cut up, okay, my linear bit of DNA. Now, if you don't leave it with the enzyme long enough, right, you might not hit all the cut sites. So let's say that should in total be 10 kilobase pairs, and I look on my gel and all the fragments, right, I've got a 5, a 3 and a 2. Well, that adds up to 10. Brilliant, they all got cut up. But there's not just one copy of the DNA. There's usually, because it's come out of PCR, multiple copies of the bits of DNA. So maybe that there didn't get cut up at all because we didn't leave it with the enzyme long enough. So all that went in the well together. So now I've got a 2, a 3, a 5 and a 10. So when I add those up, it looks like my strand was 20 kilobase pairs long, right? Which is a surefire indication. If you've got fragments that add up to more than what you originally put in, that's a surefire indication you've got some missed cut sites. We call it a partial digest. More on that again in the next lesson, okay? And different restriction enzymes will cut in the same different restriction enzymes will cut the same piece of DNA up differently, okay? So if you cut one strand up with EcoR1 and then you cut the same strand up with BAM-H1, you're gonna get different fragments because they're cut in different places. That can be really annoying if you've got 2 different labs using 2 different blends of enzymes, or it can be really good. If you cut 2 different things up and 2 different samples up with the same enzyme and they are kind of similar strands, it doesn't really give you a conclusive conviction. You're the murderer, right? It doesn't give you that. Maybe use that same DNA but try another enzyme and another enzyme, okay? So then you get more evidence because you've gone, well, when digested with this enzyme, we get this, and with this enzyme, we get this, and this enzyme, we get that, okay? Which gives you more information. So please do bear that in mind, okay? Next lesson, we're going to look into how we can apply this in the lab and indeed on an exam paper, how you can interpret these fragments on a gel, because that is a good logical thinking skill. So we'll leave it there for now and come back to that next time. Thanks very much for listening. I hope to see you again.